So without further ado, I would say we start our session, Nanocapsules and Nanoparticles modulating the immune system, Immunization for tumors, and dampening autoimmunity and allergy. I think we're going mostly to talk about the immunization stuff. And with this, I would like to start with targeting of nanocarriers in vivo and maximizing the tumor therapy effects with antigen-antigen adjuvant combinations. Next slide, please. We don't, in this CRC, in Collaborative Research Center, or Sonderforschungsbereich, as we call it in German, 1066, this was actually a long time initiated. It's 10 years now, it's going to go for another 2 years. And this was the starting point of the checkpoint inhibitor era, so to speak, but it was not yet clear what it really means. And Professor Grabbe, who is unfortunately not here, and Professor Zähnl, they agreed, well, we're going to do something with the immune system. We're not going to target the tumor directly. It was never a goal to actively target that, and it was even excluded from the SFB. So what do we target? Of course, a lot of people, when we started that, were saying, okay, you need to go for the T cells as the effector cells. But when you look at the immune reaction is orchestrated by dendritic cells. Just very shortly, first for the real chemists, I don't think there are too many in the room, but so it's the most important cell type for vaccination. It's capable of engulfing and processing the particulate antigens, the nanoparticles. Okay, it can prime T cells and induce antigen-specific immune responses, and you can do a targeted delivery of antigens or adjuvants is probably key. But it's not clear if you could just combine them into a syringe or if you really have to combine them in your nanocarrier itself. Okay, now it's working. Okay. Obviously, there's a time delay between the different ones. Come on. So, what we were looking at conventional conjugation techniques, and to make a long story short, if you try to do that with usual chemistry or with EDT, NHS chemistry, you get binding of your antibody in all directions. You get even binding with the FC part, if the FC part showing to the outwards, and then the FC receptor is really then internalizing that into macrophages and Kupffer cells. So these are not the cells you want to go into because they are dampening, in the end, your immune response. So we wanted to have a site-directed oriented or conjugation to go into different types of dendritic cells. Not going to go into the biology, and it was conventional type 1 and type 2 dendritic cells that we wanted to get into, so the chemistry is shown here. That's actually something done that Johannes, Simon, Gabor Kuhn, Max Brückner, and also Carina Jung are doing now, who is in the audience. So basically what we did is by conventional NHS chemistry, we were functionalizing the surface of the particle. That's what the chemists do, not I as a medical doctor. And then we were looking for a site-directed conjugation strategy. And this is done by using the carbohydrates on the FC part. You use a galactosidase in order to remove the galactose from the end. And then you use a transferase in order to attach UDP-GalNAc with an acid on it. And this is, so it could give you 4 sites to conjugate to, but you only need one site, and you click it. Well, it's a 24-hour reaction overnight, so it's not a very fast click reaction, but it's a very precise reaction which is going on. And so we were looking, what does it mean to antibody functionalized nanoparticles? And of course, what you see here, first we're looking in, this is actually done in mice. So we did all the in vitro studies before, and you can read about that in the paper shown below. But this is just the in vitro part of that paper. And what we did, basically, you see the magnetic hydroxyacyl starch particle on the left side, and the DBCO functionalized, IgG conjugated as all control, then CD11C as pan-DC marker, and then different types of sub-DC markers which you wanted to use as entry points into the cell. And you see, well, there's not uptake into macrophages, no specific uptake, which is nice, but of course you still have something like 30% of the cells of the macrophage which take up something because it's nanoparticles. And when you look then at all the CD11C dendritic cells, Then you see, okay, you find uptake in, of course, the CD11c positive ones. You find uptake in the conventional disease type 1, about 50 to 100% more, so to speak. Sounds a lot, but it's not so much, right? I mean, you would have to like something for an antibody, you would have something like 100 to 1 or something like that. So here it's only something like a factor of 2 at best. And also for the CDC type 2, you get something like 50% more with the pan-CD marker, pan-dendritic cell marker, the CD11c. And for the others, you're getting less, although statistically significant, but biologically the question is if that's significant. But we will also look at other cell types, and the plasma-cytoid disease, which were not our target, they were actually the best. And they have something like a 4 times higher uptake, and you get up to 70% of these cells you can really load with these nanoparticles in the end. And you can do that with the pan-CD DC marker CD11c, and you can do it actually with CLAC9A, which is nice because this is really getting you into a more detailed or more specific dendritic cell. And the good thing is, and the plasmacytoid disease, they go into CDC type 1 in the end, so they may be a good target. And I will try to tell you in a moment why I think that is. We were looking at, this is done by Carina Jung, we were looking into smaller, and we were trying to get rid of the FC parts, doing FAB parts, doing single chain. Nothing of that worked really. Then we looked for these nanobody parts, these camelid antibodies. The chemistry is given below. Carina can comment on that on her poster if you want to. It's much easier, much faster, and all the production, of course, of the nanobody is something that you could do in a much larger scale. And this, when you look at this, you don't have to look at each, it's a busy slide, but if you look at it, it's basically the same. Actually, when it was first shown to me, I asked Michael, did you, and Michael and Jenny Schunke, Michael and Jenny Schunke, did you really copy that? I mean, it's exact, it looks like exactly the same. So it is not, of course, it's a different mouse, it's different experiments, it's different particle, it's a new synthesis, but it is basically the same as results. So really this works, even with another nanobody, with another antibody format. And the big question is, why is there better targeting in PDCs, in these plasmacytoid dendritic cells? And we don't really have the answer, but I would have like to present a hypothesis, and this is because PDCs are located in the blood, while the CDC1, CDC2s are in tissue, spleen or other. So basically you have a physical border between your nanoparticle and your cell. And this is giving you less concentration of the particle, so how it's bathed in the nanoparticle solution. And probably this is even worse for every tumor for several reasons, intratumoral pressure, things like that. So what is the biological effect of a nanocarrier in tumor vaccination? Can we treat tumors with that? And is it necessary that we include the antigen and adjuvants into the same format, so to speak? Or can we do that all insoluble? Can we just put it into a syringe and inject it? Or do we have to really to make all this chemistry? And there's no targeting from now on, so don't confuse that. This is just, so to speak, the second part, not yet conjugated, not yet put together with the first part I showed you just before. We used ovalbumin as an antigen. Then we used 2 adjuvants in order to trigger the immune response. We pre-established a tumor for 6 days. So it's a fast-growing tumor model, B16 ova cells. We immunize for 3 times with a high dose, and then we sacrifice at day 90 if they ever got to that point. And what you, this is also a very busy slide, and what you can see here is basically, if you put everything together in one nanocarrier, OVA, R848, DAPC, then you get a best response, and you get something like 60% of our mice survived until day 90. You could re-challenge them. You could, they have been cured, really. If you use just DAPC, then you have something like 20%. So definitely less effective. And if you do anything of that soluble, then you reduce the effectiveness dramatically. So you don't cure anymore. This is the one thing. Ovalbumin is is not really a physiological target. So we're questioning, of course, can we do that with a physiological target, which is tyrosinase-related protein 2, TRP2, and this is only a peptide, not a whole antibody, not a whole protein, and this is less effective. So this is something where we would probably like to combine this with a targeting and then see if we can get better by targeting. This will be our, so to speak, our standard model for the future, which we have established now. And what's the outlook? Of course, we want to enhance dendritic cell uptake. We want to enhance the anti-cancer tumor immune response. And translating that from a mouse to a human will be challenging, and I'm aware that this is a really complicated system we are using here, but it's something that we think we can show what targeting does. It can enhance your uptake by a factor of 2, and if it's in your blood, it can enhance your uptake by a factor of 4. That's the most you can get, probably. And it's not going to be 100 to 1 or 1,000 to 1 like with antibodies, because you have a large thing that's going to be taken up at least from the dendritic cells, and that's going to be our challenge. I would like to thank everybody, of course, especially people in the room, and the Michael Fichter, who is not here, Stephan Grabbe for supporting us, Karina Jung, Jenny Schunke. They have been doing most of the work, they have been doing the heavy lifting, and I think we take the questions afterwards. Thank you very much.